Hello, everybody. It's a pleasure to be speaking in front of you today at MDTX, a fabulous inaugural event. Thanks, Joe. And I'd like to talk just briefly, just give you a general overview of the terminal sterilization industry, the players, and provide you with insight where the applications are best, depending on the type of medical device that you're developing. So basically, with terminal sterilization, it is defined by ISO as the definition of sterility is free from viable microorganisms. The problem with this definition is to test and to statistically verify the achievement of the condition. Thus, sterility assurance level has been developed. And basically, there's two values that are primarily used within the medical device industry. SAL levels of 10 to the negative 3 and 10 to the negative 6. And basically, that represents the probability of a unit being non-sterile out of a defined population. And for 10 to the negative 3, that represents one device out of 1,000 having any statistical probability of being contaminated. And 10 to the negative 6 is 1 out of 1 million devices having any statistical probability of being contaminated. And SAL 10 to the negative 3, it's not commonly used for medical devices. For drugs, drugs are typically sterilized 10 to the negative 3, but that is changing. The FDA is now requiring where drugs can be terminally sterilized, they want drugs sterilized 10 to the negative 6. Now, for any medical devices that would breach the skin or carry bodily fluids to or from a patient, any medical device that is a class 3 medical device must be sterilized 10 to the negative 6. And as I mentioned, most devices and combination devices are sterilized 10 to the negative 6. So with the landscape of terminal sterilization, there's many terminal sterilization modalities out there, but 3 of which are used for over 90% of all medical devices. And basically, it comes down to a gaseous sterilization method or irradiation sterilization. Most medical devices are sterilized via ethylene oxide, which is a gaseous sterilization process. And by the way, with all sterilization modalities, they all have their place. They all have their disadvantages and advantages. And they all are the optimal sterilization modality depending on the medical device to be sterilized. With ethylene oxide, it is, again, the most commonly used. And you would want to use ethylene oxide if you have a medical device that contains any active electronic components, memories, semiconductors, or if it contains fluoropolymers such as PTFE or Teflon or acetal. Now, the drawback with ethylene oxide is that it is a very noxious, toxic process. There's an in-gassing and out-gassing process that could be a very elongated process depending on the complexity of the device. And also, ethylene oxide is a known carcinogen. And however minuscule the residuals that are left on the device, it's a big concern. And it's been rising as a concern within the medical device industry, certainly over the last decade. And even with the EPA, prior to the current administration, they had proposed lowering the acceptable minimum level of residuals of ethylene oxide on products. Now, obviously, with now, times are changing within the EPA, and also that would have a major impact on the medical device industry if that had to be the case, because all medical devices would have to be revalidated, and that would be a tremendous undertaking. But ethylene oxide, it is the most widely used. Probably more than 50% of all medical devices are sterilized via ethylene oxide. It's very effective and it's very cheap. But there is a groundswell occurring with the concerns of ethylene oxide. Then you have gamma, which is probably perhaps like 30 to 35% of all medical devices are sterilized via gamma. Another very effective sterilization modality. And with gamma, basically what you're dealing with is radioactive material, cobalt-60. Product is in a chamber, and a grid of cobalt-60 nuclear rods is in a grid array kept underwater, and then it's brought above when it's ready to sterilize the medical devices. And basically, the mechanism of gamma irradiation, of gamma is photons, is the mechanism of ionization that disrupts the DNA and RNA of microbes, rendering the product sterile. Gamma is it's very good when you're dealing with dense product in dense configurations. And it is, like I said, perhaps like 30 to 35% of all medical devices are sterilized with gamma. Then we have E-beam, which is another form of irradiation sterilization. But with E-beam, the mechanism of irradiation is high energy electrons or beta particles. And basically, you use an electron beam accelerator, and it's a beam of high energy electrons, literally somebody waving a flashlight up and down with a beam of high energy electrons that goes right through the product, right through the boxes. And just like with gamma, it disrupts the DNA and RNA of microbes, rendering products sterile. Now, E-beam is just a smaller percentage compared to gamma and ethylene oxide, but it's emerging as the practical application for more complex devices and devices that contain sensitive materials, particularly when we're getting into drug-coated stents, combination devices. E-beam is the superior sterilization modality because of the flexibility that it offers, and it's less harsh to product than gamma. There's a direct correlation between the damaging effects of irradiation and the time that the product is exposed to that source of irradiation, regardless of the source. And the deleterious effects that occurs to the product is primarily due to oxidation. So removing oxygen within the packaging environment usually has a big impact on optimizing the post-sterilization performance of the product. So E-beam, for example, if the product contains drugs or drug products itself, allograft tissue or biologics, you really want to take a close look at E-beam. Now, within the terminal sterilization industry itself, basically, this industry is a duopoly. It's controlled by two companies, Steris and Sterigenics, very good companies, very knowledgeable. And even though that the industry is a duopoly, essentially the medical device industries needs these large players with global infrastructure to serve their global needs. And both Steris and Sterigenics have made incredibly smart acquisitions over the recent years. Now, it has raised questions about the vertical integration of these industries and how much control that they have. For example, Steris, they purchased a European sterilization, contract sterilization company, Synergy, and so that basically gave Steris a global footprint. Very smart acquisition. It did raise some concerns initially with the FCC, but obviously it worked out, and now it's integrated as part of Steris. Sterigenics, some notable acquisitions that they made is with Nordion in 2015, and Nordion is the sole supplier of cobalt-60 to the gamma irradiation sterilization industry, which in the Western Hemisphere, there's only two players, Steris and Sterigenics. So they purchased Nordion in 2015. So basically, Steris has to purchase cobalt-60 from its arch rival, Sterigenics. And also last year, or in 2016, Sterigenics, they purchased Nelson Labs, which is considered the premier microbiology testing lab, certainly within the United States. So, and then that's being integrated into Sterigenics at their local, local facilities. They used to have a microbiology testing lab called SteriPro, and they're rebranding that as Nelson Labs due to the high reputation that Nelson Labs has. So excellent acquisitions by these companies. They know what they're doing, and they're only becoming stronger. And there's also other sterilization, terminal sterilization methods that are worth noting, particularly Revox and Noxalizer. Revox, they've developed a chamber for VPA, vaporized peracetic acid. That's a very interesting technology. As our company, Steritech, we're very interested in this technology. By the way, Steritech, and I'll get a little bit more into this, we're an E-beam X-ray player, but we're basically agnostic when it comes to novel sterilization approaches, and we want to embrace whatever we consider to be the future of where the terminal sterilization industry is concerned. And we're certainly willing to accommodate companies such as Revox and Noxalizer to partner up with them, and even we've offered to have systems at our facility to help promote their technology. But Revox and Noxalizer, which is vaporized peracetic acid and nitrogen dioxide respectively, they have advantages over ethylene oxide because product could be processed at room temperature. Whereas with ethylene oxide, you're typically looking at a minimum processing temperature of 50 C. And for sensitive materials, it could be too deleterious to sterilize via EO. So those are interesting technologies. And other sterilization technologies, there's autoclave or dry heat, steam, plasma, pulse light, microwave, so sound waves. So these are all interesting technologies, but none of these technologies here are used on a mass scale. Revox recently has been approved for their chamber. It's been approved by the FDA, so it's recognized as an acceptable terminal sterilization method. However, they just right now have like in-house chambers, so you cannot sterilize on a large scale. But regardless, interesting technologies to keep note of. And so basically with Steritech, we're just, compared to Steris and Sterigenics, we're just a small player. We're referred to in the industry as a boutique or a niche sterilization company, and we accept that label with honor because we are a boutique sterilization facility. We focus on high-value medical devices and sensitive materials. So combination devices, drugs, allograft tissue, we are heads and shoulders above our competition insofar as collaborating with our customers to prove out E-beam, and once that's proved out, to optimize the sterilization performance of their product. And we do work with our customers on a collaborative level, and with our predecessor company, we've developed a lot of the advanced methods for E-beam sterilization processing that is now used globally. We were actually formed, Steritech, we've been in business since April of 2016, so we're relatively new, but we're not new because we were actually a company prior to Steritech, NewTek Corporation, since 1990, which started out of Palo Alto, California. We worked in conjunction with Stanford in developing E-beam technology, and what happened, we were a flourishing company, but we were a victim of eminent domain with BART, which is the mass rapid transit for the San Francisco Bay Area. So basically, we're the same faces, but a new name. And as I had mentioned, we're the innovator in E-beam sterilization for complex medical devices, drugs, biologics, and allograft tissue. Our core team has over 75 years experience combined within the E-beam industry. We are a pioneer with X-ray sterilization. In fact, we are the only X-ray sterilization facility in the United States. And we are the only E-beam facility in the world with the DEA that can process controlled substances for drugs that are Schedule II and higher. So, And again, we're based in the San Francisco Bay Area with plans to build a new facility east of the Mississippi River. And so basically with E-beam, whereas with gamma, product is in a chamber absorbing radioactive energy perhaps 6 to 8 hours, depending on the type of product and also the energy in the nuclear rods. With E-beam, it's very fast and efficacious. So the boxes, they go on a tote, and these totes, they travel through an E-beam bunker, which is 10 million pounds of concrete. And then with Steritech system, we have a dual beam system. We have two 10 MeV, 20 kilowatt accelerators. And we take it as a compliment because Steris has built 2 E-beam facilities in upstate New York and in Los Angeles, and they've replicated our design. And we take that as a compliment. So basically, with our facility, with the totes, they travel through the E-beam bunker. They'll encounter the first of 2 E-beam accelerators, and they get E-beamed on one side of the box, and they're in front of the beam perhaps 30 to 45 seconds versus gamma 6 to 8 hours. So there's less time for oxidation to occur. So that's why the post-sterilization functionality or performance of products sterilized by E-beam is superior to gamma. So with our dual beam system, the totes will travel in front of one E-beam accelerator and get dosed on one side, and then travel about 10 feet further and get dosed on the other side. And basically, once it comes out, it's a parametric release or dosimetric release. Hypothetically, the product that is processed there could go directly into a surgical suite and implanted into a patient. And then just some of the advantages of E-beam and X-ray. It's FDA approved, just like E-beam or gamma ethylene oxide, and now with VPA. Now, the electron accelerators, that's an on-off technology, so there's no residual radiation. And it delivers the dose rapidly, which protects the product's function. And very importantly with E-beam, the processes are flexible. They're easily specialized and easily validated, including continuous temperature control, split dose processing, intelligent tooling, etc. And it causes no damage to the seals or packaging. And we're just an overview of our certifications. We're ISO 13485, ISO 11137. We're DEA approved for Schedule II drugs and above. We're FDA medical device, tissue and biologics and tissue registered, as well as FDA drug registered facility, and also State of California medical device, the State of California drug, as well as we're registered with the Japanese Ministry of Health for PMA devices. So, and then just some of the additional capabilities. We have walk-in freezers for drugs. We have a very controlled process for securing controlled drug products with drug cages, which is required by the DEA, and a safe, a dedicated safe for Schedule II drugs. So now, very importantly, with considerations for developing medical devices, it's so important to prove out whatever sterilization modality that you're considering is to prove out that that modality is suitable or compatible to your product. With our history, we've witnessed companies make a fatal mistake presuming that they're going to sterilize their product with a given sterilization modality. They invest millions of dollars, go through multiple rounds of funding, determined or set on a given sterilization modality, only to find out when it's too late that that sterilization modality is not compatible with their product. And it's incredible that these smart people would make such a fatal mistake, but we've seen companies go out of business because of this fatal error. So I cannot stress enough with developing a medical device to prove out that sterilization modality, and once that sterilization modality is proven out, to optimize. And it doesn't matter if it's ethylene oxide, gamma, or E-beam. Now, when it comes to irradiation or terminal sterilization, also for E-beam, you're going to look at special techniques and intelligent tooling, whereas E-beam offers that flexibility. And also an important consideration for validating your product is the level of bioburden on the product, or basically how clean your manufacturing process is. So for irradiation sterilization, regardless if it's gamma, E-beam, or X-ray, the level of bioburden will determine the dose that your product can be validated at. So for sensitive materials, this is extremely critical. So for example, most devices can be relatively dirty and be validated at 25 kilogray, which is most commonly the dose that products are validated at. But if your product has on average for 10 devices, each from 3 separate lots, a measurement of CFUs or colony forming units, if your product has on average 45 CFUs or less, you could validate your product at 20 kilogray. And if your product has, for example, 9 CFU or less for 10 devices from 3 separate lots, you could validate your product at 17.5 kilogray. Now, being able to lower the validated dose for your product is absolutely critical with sensitive materials, because there's going to be a dose range that a typical configuration will receive regardless of what it's validated at, and to shift that down could make all the difference in the world if the product can be sterilized via, in this case, e-beam. And also considerations, you do want to look at the regulatory landscape, the landscape of the sterilization industry itself. And also, kind of a big picture type of thing is environmental concerns. With ethylene oxide, it's a very noxious process. Not only is it a known carcinogen, but it's a pollutant to our atmosphere. With gamma, we're dealing with nuclear rods that, for the Western Hemisphere, it comes out of Canada, distributed, like I said, with Nordion. And with gamma, this is radioactive material that once the energy is used up, they have to go into these elaborate containers and then get buried into a nuclear waste site, of which there's 70 nuclear waste sites in the United States. And I even read an article last year that they had proposed digging a tunnel 7 miles underneath Lake Huron to dump more nuclear waste. And we could say, well, what big deal is it? We're not going to have to it's not going to affect us in our lifetime. But what about 200, 300 years from now? Cobalt-60 takes 2,000 years to decompose to nickel. So that needs to be looked at, the big picture. And that's what's happening with the groundswell now, is companies are looking for alternative methods to ethylene oxide and gamma, and e-beam is emerging as that application. So with that said, I just made it just under the time.
You did
I want to
a
thank you again
fantastic job.
for this fabulous...
Jeff, that was a solid presentation, and I'm singling you out, but really, thank you to everyone who has taken the stage so far. I think you'll agree, we have not had one bad speaker. Everyone has delivered solid information and value. Thank you very much.
You're welcome. Thanks, everybody.